Hi, everybody. My name is Claudia Camerino. I'm Associate Professor at the University of Bari School of Medicine, Department of Pharmacology, and I conduct basic and translational research on the hormone neurotransmitter oxytocin, investigating the role of oxytocin in obesity, metabolic disorder, muscle strength, bone biology, and we apply this knowledge to several diseases like Prader-Willi syndrome, Schaff-Yang syndrome, autistic spectrum disorder. Today, I'm glad to introduce the recent work by Dr. Yi Nan, titled, I have this paper here, I have to read the title, Dynamic Changes in Ubiquitous Glycosylation Regulate Osteoclast Differentiation and Bone Loss via Nucleoporin 153. Dr. Yi Nan is currently finishing his PhD in biology and is starting his own research group in HubLE Research Center, Department of Rheumatology of University Hospital in Düsseldorf, Germany. In this study, Dr. Li shows that osteoclast differentiation requires a dynamic regulation of ubiquitous glycosylation with increasing levels during early stages and progressive decline to baseline levels in late stage of osteoclastogenesis. So now Dr. Li will introduce himself and he will tell us how his work fits in the big picture of the field. Please, Dr. Li.
Hi, I'm Nan. Thank you very much, Claudia, for the detailed introductions.
My honor.
I also want to thank HubLE for inviting me and their interest in our work. So I'm Nan. I'm a postdoc at HubLE Research Center at Düsseldorf, Germany. So in this work, we look at the function of OGNAC, glycosylation, on the differentiation process of the bone eater cells, osteoclast. So we found that this OGNAC can promote the differentiation of osteoclast. However, the downregulation of OGNAC is also important for the cellular maturation. So I believe both process is important for the formation of functional osteoclast. So this will have further impacts on the bone erosion or bone loss in arthritis or diabetes as well.
Dr. Li, I have a few questions for you. So first question, what do the results of this work mean to you?
So I am pretty amazed about our findings. So basically we found this metabolite is a sugar, right? Glycosylation. Attachment of sugar and the protein can have this big impact on the fate and activity of the cells. So these findings will definitely invite other researchers in the field to look into more metabolic status of the cells or even at tissue level when they approach this topic.
Another question for you. What is the most important thing you learned from this study?
Well, the most important thing we learn is that to always keep the open mindset and curiosities throughout research. I know it sounds pretty cheesy, but we found OGNAC has both promoting and inhibitory effects on osteoclast formation. This might sound confusing at first. However, when we sit down and look at the data, we found distinct functions or quite opposite functions of OGNAC at different cellular state or different time points. So I think embracing this open mindset is truly important for us to generate data-driven hypotheses.
It's called correct interpretation of data, and it's essential. So next question, what were the greatest challenge you encountered with this work and how did you overcome them?
Well, we encountered several challenges during research as always. I think the biggest one is to figure out how we can handle this omic data. So in this study, we implement RNA sequencing and glycoproteomics to look at the changes in transcriptome or OGNAC associated protein. Both methods are quite new to me, so I think the key challenge is to figure out how we can extract insight from these large data sets. To overcome it, I think it was a team effort. So I think communication and teamwork is key. We have discussions readjusting our aim, communication with bioinformaticians, I think at the end, it works out pretty well for us.
That's very good. Next question. What was your experience publishing in bone research?
Well, as this is the first article, I'm in a first position. So this is a really...
Congratulations.
Thank you. This was really valuable experience for me personally. The comments from the reviewers were fair and insightful. They do point out some blind spots we did not intensively address in the initial submission. However, after this peer review process, we have included all the insight we got in the article for the community.
Very good. Next question. So what do you expect the outcome of this work to be? Further research, trial, patient work, bench to bedside?
Well, I think for follow-up studies, definitely. So in this work, we focus on the function of organoid in osteoclasts because they are the bone eater cells. They are effector cells to erode the bone. However, I think its functions of other cellular component is also important. So I believe after we get the comprehensive view of how organoid behave in the tissue level with all type of cells, then the clinical translation is definitely coming next, I believe.
Last question, and in terms of your career and future goals, how will this publication help you to achieve those aims?
Well, first, I think this publication serves as a recognition from the expert in the field, which is a confident boost for me as I'm quite junior. And furthermore, this also showcases our capability in the technical wise or topic wise. This will come in handy when we want to secure more fundings. So I believe this publication do help me to grow as a more mature researcher.
This is very nice to hear. So, I personally, I read this work, you see my paper, my note on the side, and I personally like this study because it can be applied to inflammatory model of bone loss, like rheumatoid arthritis, or also to non-inflammatory model because ubiquitous glycosylation is involved in bone loss in ovariectomized mice. So my last two questions are, do you think these studies will have a translational meaning in rheumatoid arthritis and osteoporosis? And how long did it take you to complete this work? Because I also think this is your PhD thesis.
Okay, so for the first question, yeah, both. I think because we see the beneficial effects in both inflammatory arthritis and osteoporosis mouse models in this study. However, we do see inflammatory stimuli can boost the organoid level in cell culture. So I personally think that targeting organoid can have even more beneficial effects in rheumatoid arthritis. Second, regarding the timeline of the study, yes, this is my PhD thesis, and I was really fortunate when I joined. This project is already in progress. So afterwards, we spent around 2 to 3 years to dive into the mechanism of how this attachment of sugar can lead to pathological bone loss. So for my involvement, it took us 2 to 3 years, but for the whole project, it's a big one. So it takes more than 5 years, definitely.
All right. So Dr. Li, thank you very much for your interview. Congratulations for your work. The best of luck, also because I think a junior colleague must always be encouraged. So thank you very much.
Thank you as well. Bye-bye.
Ciao, ciao.